All right, a real short unit for you. I want to go over the production of antibody, which is our third line of defense. This is a cooperation of T cells or lymphocytes and plasma cells, B cells, that together recognize foreign antigens, directly destroy those cells, and produce antibodies as plasma cells. This cooperation of T cells and B cells results in the production of antibody. Okay, so there are actually 4 parts to the production of antibody, and you're going to want to know all 4 parts, okay? We've got the macrophage part, which is step number 1. That's the macrophage part. And then we have the second part, which is the T cell part. This is called T helper cell sensitization. And then we have the third part, which is B cell part, which is B cell activation. And finally, the fourth part is the production of antibody is terminated by the T regulator cells. So let's go talk about the production of antibody. We'll outline all 4 steps. So step 1 in the production of antibody is the role of macrophages, okay? Macrophages actually process the antigen by phagocytosis and degradation. Meanwhile, selected portions are conserved for presentation to the T helper cells. So antigen is presented to responding T cells and some B cells. Thus, T and B cells work cooperatively to make antibody. Interleukin-1, which is also known as lymphocyte activating factor, is secreted by the macrophage, which activates T helper cells that were previously resting. The macrophage makes a monokine known as interleukin-1, and that is secreted by the macrophage. Step 2 in the formation of antibody is called T helper cell sensitization. It is the role of T lymphocytes that provide help for antibody production by B cells. The T helper cell binds to the antigen in the macrophage, and this stimulates the production of lymphokines. One lymphokine that is quite important is interleukin-2, which is also known as T cell growth factor. Interleukin-2 initiates proliferation of activated T cells. These activated T cells produce many T helper cells, and these cells are suppressor and cytotoxic-like memory cells. Interleukin-2 is the lymphokine secreted by T helper cells, and though that is called T helper cell sensitization. Step 3 in the formation of antibody is B cell activation, and here we see B lymphocytes play a role in an immune response. Once T helper cells are sensitized, B cells bind to T helper cells and the macrophage and the antigen. This is called a tricellular complex of T helper cells, macrophage, and antigen. This tricellular complex of antigen, macrophage, and T helper cell activates the B cell, which generates receptors for B cell growth factor. B cell growth factor makes plasma cells and memory cells, and also B cell differentiation factor. This is the factor that directs plasma cells to produce the individual antibody classes. Okay? B cells reproduce growth factor and make plasma cells, memory cells, and immunoglobulins. B cell growth factor causes proliferation of the B cells, plasma cells, and memory cells, while B cell differentiation factor directs plasma cells to produce the individual antibody classes. This brings us to the final step in antibody production. The final step in immune defense system, the final step in the antibody formation, is when antibody production is terminated by the T suppressor cells. And those T suppressor cells are now called T regulator cells. So make sure that you know all the steps in the antibody formation.